첫 번째 발
Yes, that was the first presentation, but this was a recorded tape, and so we will not have a separate Q&A session, and we will go right into the second presentation, which will be on combating AMR, an overview of bacteriophage therapy, and Professor Yu Sang-yeol of SNU will be giving this presentation. Professor Yu received his degree, bachelor and master's at Seoul National University, and also he received his PhD in food microbiology from the University of Massachusetts. Let's welcome him with a big hand.
예, 감사합니다.
Thank you very much. So today, during this talk, or during this session, we're trying to utilize different methods in order to deal with the issue of AMR. So there's a lot of recent technology. And recently, there's a lot of interest in bacteriophage therapy, and that is what my talk is going to be on. As you all know, at a global level, we have different antibiotics, and we are seeing there are organisms that have developed resistance. And there is more concern, bigger concern now, because we have been using more antibiotics because of the COVID-19 pandemic. So this means that there will be more organisms in the future resistant to these antimicrobial agents. So one solution could be bacteriophage. So this is a type of virus. However, it infects only bacteria, and it is harmless to human cells and also animal and plant cells. And in the world, on the globe, there are 10 to the 31st power to 32nd power macrophages in the world. So it's very, very abundant. And by these bacteriophages, 50% of the bacteria that exist die in a day, and then they regenerate. And there are two cycles in the bacteriophage life cycle. And so the bacteriophage goes into the host, it kills the host, and then it comes out. And then also it can go inside the chromosome of the host, and it will be part of the chromosome. So that's part of the life cycle as well. For therapy use, we will be using the lytic cycle that you see on the left side of the diagram. So the lytic cycle. And if you look at the genome, you can see that it has great diversity, ranging from 17 kilobase pair to 500 kilobase pair. And bacteriophages are very, very numerous and high in number. And so even if we analyze the smallest gene, there are some genes that we find where we cannot find out the function, and that's about 15% of the total genes. And so this means that this can provide new genes, novel genes for numerous applications. So this is history. So we found out about phages in 1890s to 1900. So there was this person called Hankin, and he observed antibacterial activity against Vibrio in the Indian river water. And after that, it was confirmed that bacteriophage is effective in dealing with bacteria. And before we had antibiotics, we were using bacteriophage to treat certain diseases, certain infections, and it was used as a panacea, and it was commonly sold in drugstores and pharmacies. However, there was a problem. because at that time, people did not know that bacteriophage had specificity for bacteria. So sometimes it did wonders for the disease, sometimes it did nothing. And then since the 1940s, antibiotics were developed, and in the West, antibiotics were used. So bacteriophage use decreased drastically. However, in Soviet Georgia or in the East, there was continued research. And you can see that we have a research institute that has been established, and they celebrated their 100th year this year. However, now we have the problem of antibiotic resistance and now renewed interest in bacteriophages.
6년
And in year 2006, US FDA approved the use of phage cocktail for the use in RTE meats to prevent listeria. Now, there are two characteristics of phage. The first one is that it shows highly specific infection. And we have metagenome, where there's a lot of interest here. And there will be no issues raised in the microbiome, and it can target the specific bacteria. So that's one big advantage of bacteriophage. Another thing is that when it goes inside the host, it amplifies tenfold to 1,000-fold. We can use this characteristics, and we can detect a very small number of pathogens by using macrophages because they amplify so fast. And so how can we use these characteristics for therapeutic use? So that is where we will have to do future research. So we have the phage, but we have other things that we have to consider as well. We have to think about lysis, which has to happen. And lysis, well, we have the lysin, which is the enzyme that is created by the phage, and this is degraded by glycan, and we can use the phage for therapeutic uses. But not only that, we have endolysin, which is a byproduct of this process, and we can clone this outside the cell, and we can use this material in order to break down cell walls, and that can be used for therapeutic uses as well. So I would like to talk about phage today, and also endolysin, how we can utilize these two different materials. So in this table, I compare antibiotics and the bacteriophages and the endolysin. So basically, for antibiotics, specificity is very, very low. And so if you give antibiotics, most of the bacteria will die. However, there is specificity for the phages and the endolysin, and so we can minimize side effects. Also, the system is quite simple. It's very easy to develop. These are some major characteristics. Moreover, we may have resistance against bacteriophages as well. However, the bacteria that develops the resistance has very little pathogenic characteristics. And so receptors are used to enter cells, and these receptors play a role so that the bacteria that has resistance against the bacteriophage has lost part of its component, and it is no longer so pathogenic. And so when we use phage, there could be some resistant bacteria. However, it's less pathogenic, so it may not be a big issue. So that's a very big advantage. And so as you can see here, many companies are working on the development of bacteriophage in order to develop drugs and treatments. So first of all, let me explain phage therapy. So as I said, there are diverse phages, so if we play the game correctly, then we can target the exact bacteria that we do want to target. And so here I want to talk about the Salmonella phages that we are studying at our lab. So we have isolated dozens of such phages, and there are short ones, long phages that is. And then there are ones with tails that contract. So there are three major categories. And so in our case, well, the bacteriophage will go inside the host. So we did a lot of study on the receptors. And the host range is quite narrow, so we mix the phages, so we call them phage cocktails. And usually we mix 3 to 4 different types of phages, and we try to control specific strains of bacteria. Now, in order to mix the different types of phages, well, maybe we can utilize phages that have different receptors. And when we have resistant bacteria in the future, well, we can lower the possibility of that happening by having these different receptor types in one single cocktail. So we have TN5 random mutant library. We have utilized that. And we screened the receptors of the phages that we found. And the result is what you see here on this table. So we looked at the receptors of 20 plus phages. And as you can see here, it's quite interesting because they roughly fall into 3. So it's either Flagella or BTUB or LPS. These were the 3 different receptors that were being used. So 3 groups existed. And so in each group, we did a bacterial challenge assay. We wanted to pick the one that did the best job in killing the bacteria. And by using the 3, combining the 3, we came up with a cocktail. And the morphology of the selected phages are as you see on the screen. Now, this is the genome sequencing that we did for the 3 different phages. And we looked for specific pathogenic genes or genomes, or we made sure that they did not have the genomes that we did not want, and that they are indeed the right candidates for the cocktail that we're trying to make. And so we selected the 3 bacteriophages, and you can see the host range analysis that we did for the 3 phages. And we test for Salmonella, and you can see that all 3 were effective. And as for BSP-101 phage, it's quite unique because it works for Salmonella, but also E. coli and also O157 pathogen. It was quite effective in controlling all 3 of these pathogens. Now, as for BSP-101, we have the O antigen. So it uses the receptor that the BSP-101 uses is the O antigen. So judging from that, well, maybe it has to do with the O antigen structure of the Salmonella. It's quite similar, and that's why it's effective. So we chose the 3 phages, and we did the bacterial challenge to check out the effectiveness. And here you can see the efficiency of the 3 phage cocktail. And so you can see the control and the individual phages working, and then you can see the efficiency of the cocktail. We also looked at how often mutants are likely to occur. So in the 3 phage cocktail, you can see that compared to mixtures of 2 or just single phages, you can see that the BIM is actually lower. And also we took iceberg lettuce and cucumber and did actual exams. And you can see that there is a reduction effect of more than 5 logs. And we isolated 2 other phages and we mixed them together. And we did some mice experiments. And usually the mice, well, we have salmonella infection, and usually they die within 10 days. However, we had MI10, so multiplicity of infection of 10 is the infection that was given. And so that is the ratio between the bacteria and the bacteriophage. And we used the infection factor of 10, and only with that you can see that it was quite effective against salmonella, and the mice was perfectly protected from salmonella infection, and it was able to live. And we can think of applying this to food and also mice, and you can see that in the past 2 experiments, our cocktail is very effective. So as you have seen thus far, there could be numerous applications for bacteriophages. And during what I talked about, well, I talked about narrow range of host specificity. This could be an advantage. In some cases, if we cannot find the right host range, then we cannot use the phage for therapeutic effects. So perhaps we can manipulate and we can do phage engineering to make that match. And maybe we can extend the host range of phages. So we did an experiment on that, and I would like to share the results. So generally, we have the tail spike protein in the tails of phages, and they actually determine the host range. And so we looked at the numerous phages that we had, and we tried to substitute the different tail spikes in the receptors, and we looked at how that changed the host range. And so this is the tail spike protein of the phages that we have, and we have these results, and then we also have other published information from other labs. And amino acid sequence residue that was in the range of 400 to 450, and they played a very important role in interacting with other proteins. And so based on that information, this is what we did. So we have P22, TS3, T56, 32H, and so forth. So we looked at the tail spike protein in these tails, and we used CRISPR-9 technology, and we came up with this hyperphage. And so the protein sequence from P22 was switched with the tail spike protein in the receptor of 32H and so forth. So we did switching of the tail spike protein, and we have compared the wild type and the mutant. And the marker that you see here on the bottom, you can see that there was structural change to the genes, and you can see that specificity changes following the switches that we made. And the LPS structure changes, and accordingly, the host range changed as well, and we could manipulate it in the direction that we wanted. And here, well, you can see that there are many different organisms that are resistant against antimicrobial agents. And so we work on choosing the right media, and we also are having difficulty in isolating our target bacteria. So Campylobacter jejuni, in that case, they have resistance. And so here we need selective media. So we have Bolton broth and Proston broth that we used. And when we put in different antibiotics, well, we can see other bacteria contaminating the broth, and we can actually work on these other contaminating bacteria in order to kill the Campylobacter jejuni. And so even when we use antimicrobial when we use antibiotics, we can see that many of the organisms in there do not die, and it is very difficult to isolate the target bacteria. So that is an issue that we are experiencing recently. And so here we're doing higher Campylobacter jejuni isolation in the presence of phages targeting E. coli. So we want phages that can kill the E. coli so that we can better isolate the Campylobacter. And we compare the efficiency in isolating the Campylobacter, and we tested it in 2 different temperature ranges. So the gray bars show the phage cocktail. So it's not 100%, but compared to the control where we did not have the phage cocktail, you can see that when we do use the cocktail, we have higher efficiency in isolating our target bacteria. And so if the sample is contaminated with these other resistant bacteria, well, then we can utilize phage cocktail to kill the non-target organism in order to isolate our target bacteria easily. And recently, well, of course, we have tried to find alternative to antibiotics, but that is not easy. And so we thought of maybe using phages and antibiotics together. And if that is more effective, perhaps we can use less antibiotics. So we have tetracycline, ampicillin, and so forth, so commonly used antibiotics. And then we combine that with phages and then try to see how effective that was. And we could not really see a clear trend there. However, BTUB, when phages use this as a receptor, when we use kanamycin at high dose, you can see that it was effective and the bacterial level did go down. And so perhaps we need to do more future research to think about how we can use phages and antibiotics together to have better effects. Next, I would like to briefly go over the topic of endolysin. So after the phages grow in number inside the cell, it tries to break out and they produce endolysin. And we can actually go through this process outside the cell wall, and it is effective, and it could be an alternative to antibiotics. So basic structure of endolysin. So we have 2 domains. They are linked together. One is the enzymatically active domain, and the other is the cell wall binding domain. As for the latter, it is more effective than antibodies, and it binds better to cells and cell walls. We can use this in order to come up with treatment. So it is a very unique enzyme. And so, for example, for S. aureus, we have lyse SA97 that works on that. This is a endolysin. And when we isolate and do the treatment, as you can see here, it is a concentration or dosage dependent result. And what is important here is that we have S. aureus pathogen. It is a serious issue because they form biofilm. And when we apply antibiotics, when we apply drugs, it cannot enter the cells. It cannot show effect because it cannot go through this biofilm. However, here, as you can see, lyse SA97 effectively disrupts the biofilm that is created by S. aureus. Also, we have carvacrol. This is a naturally occurring antibiotic, and we can combine this with lyse SA97. And we looked at the synergistic effect that it brought. So we have, you can see the effect when they are used as single agents. However, when we use 1/10 of the two in amount, combine them, and then use it, they have the tenfold effect of using the ingredients as single agents. So if you look at SEM photos, you can see that the cells of S. aureus are effectively deformed by using this cocktail. And at this time, 100 S. aureus endolysins were studied, and the diversity was not really high. We could see that roughly they could be divided into 6 different categories. And so we have the cell wall binding domain and the enzymatically active domain. That was quite unique because in type 1 and 2, they only had one. And then if we look at the other endolysins in the enzymatic active zone, it exists as singles. However, from type 3 to type 6, we can see that there are 2 enzymatic active domains. So that was quite unique. And so we had an idea here. From the perspective of the bacteriophages, there could be endolysins that are very effective, and that is sent out from the bacteriophages, and that could kill off all the bacteria that it might actually need in the future. So in nature, I believe that the bacteriophages, they exclude endolysin, which has a lesser effect, so that they can survive in that environment in the future. And so in the lab, we did in vitro tests. And perhaps it is highly likely that we can enhance this less than 100% effectiveness. And so we have numerous domains of the endolysin, and we did random shuffling of them, and we switched the positions around. So in nature, compared to what is in the nature, we are trying to find endolysin that is more effective. So we used a 2-vector system to do the expression, and then we did some screening. So we have a system of doing that. And as you can see here, what you see that is kind of black is the size of the inhibition zone is quite high when it shows up in dark color. So we can use this, and we can do the domain shuffling randomly, and then we found the ones that are more effective. And one that we found to be effective is Cli-C, and the mother form of Cli-C would be Lys-SA12. And we use 2 concentrations here to do this test, and in both concentrations, Cli-C was more antibacterial in its effect compared to Lys-SA12. And some characteristics of Cis-C. So temperature, in very high temperatures, not much of a difference, but at about 45 degrees, when we use it for therapeutic purposes, we can use it at 45 degrees for humans or animals. It was quite stable at these temperatures. And then pH was these levels. However, NaCl, you can see that Cli-C at certain levels had less stability compared to Lys-SA12. And the biofilm disruption capability of Cli-C was higher. Also, we did tests in milk, blood, and serum. And in all 3 cases, you can see that Cli-C always was the more effective compound. And where we used Lys-SA, and we did some mice test, and you can see that we gave 10 milligram per kg of the mice. We used the Cli-C, and you can see that when we used it at that dose, it provided ample protection of mice. So we isolated different types of endolysins, and we have a certain bottleneck. because the problem is, in order to isolate the endolysin, we have to isolate the bacteriophages first, and that can be sometimes easy, sometimes difficult. And so now we have a recent technology that has been developed, and we utilize the shotgun metagenome mining method. And we can use this to perhaps isolate endolysins that we were unable to isolate before. And from there, we can combine different DNAs and then come up with endolysin genes and genome that we were unable to isolate before. So we have these different types. Some work on Salmonella, some work on E. coli, some work on S. aureus, and so forth. So they show different characteristics. So by utilizing this technique, I think that we will be able to isolate more endolysins in the future. And lastly, I would like to talk about this. Endolysin that we have isolated before usually worked for gram-positive germs. However, for the gram-negative, they have this membrane, and it is difficult for the endolysin to reach the gram-negative bacteria because of the outer membrane. Now, that is why we need some membrane destructing or destabilizing material. And so we're trying to use endolysin along with peptides that can destabilize the membranes. And of course, we have many genes within the bacteriophage where we do not know the characteristics. However, there are hydrophobic proteins, and there is some likeliness or some possibility that these proteins will be able to destabilize the membranes and enter the area of interest. So there are some possibilities that we see here. And here we did some fusions and we got some clones and we're carrying out experiments. Also, there is something called sensitizer peptides. So there are certain peptides that have that membrane disrupting capability, and then we can fuse that with endolysins for better effects. And you can see that the antibacterial activity of the fusion endolysin is quite effective. So here we're using insects in order to study against E. coli and also the Acinetobacter bacteria. And also we have something called holin or phage holin. And we saw some antimicrobial effect from that as well. And also the phage holin showed different effects. Some were very effective against these different types of bacteria. So I introduced you to different research on bacteriophage and endolysins, and perhaps in the future we can utilize this technology and study as a alternative and also supplementary method to dealing with the issue of AMR. So on that note, I would like to conclude my talk. So acknowledgements, students from our lab, they're working very hard, so I want to thank my students. Thank you.
네, 감사드립니다.
Thank you very much, Professor. Well, time is limited, so I think we have time for maybe 2 questions. Any questions or comments from the floor? Yes, please.
Thank you very much for a really interesting presentation and an extensive one. I'm curious to know, in the case of Korea, how many of these products, if any, are licensed already for use in either in food for decontamination or as medicinal products to treat disease in humans or animals? Do you have already any examples? And if that is not the case, could you tell us your views about potentially challenges in the regulation of such products and what can be making difficult to bring to the market these solutions? Thank you.
nothing is licensed in Korea yet. So one phage product is licensed to be used as a feed additive, but nothing more. And I think Korean FDA should be more brave to give a license to this kind of products. But Korean FDA is too conservative, so it's not easy to get a license. Okay. So,
thank you for that great presentation, Jeff Lejeune from FAO. First, I'll disclose my bias. I am a phage proponent. I like the idea. We've got some work to do. But people who are against it argue two main things. One, that bacteria will mutate and then the phages won't work. Okay, and I don't have a problem with that because phages replicate a whole lot faster than bacteria and they mutate a whole lot faster than bacteria. So it's a cat and mouse game and the phages can catch up with the bacteria. The other one though, and I struggle with this one, is how do we make sure that the phages don't disseminate antimicrobial resistance genes? Because they can pick up genes and transfer them, and we could be acting counterproductively, trying to kill the bacteria, but instead enhancing them.
okay. For your first question, bacteria can be resistant to any phage, but as I told you, the resistant bacteria normally lose its virulence. So the bacteria resistant to the bacteriophage normally cannot cause any disease. And we make phage cocktails to reduce the probability of resistant mutation. So that if you mix 4 or 5 phages together, you can reduce the chance of resistant development very efficiently. And for your second question, bacteriophages have 2 life cycles. So for the gene dissemination, the lysogenic cycle is very important. So let me show you. Can I see my slide again? Or? Okay, this is the 2 kinds of bacteriophage life cycle. So when the bacteriophage use this lysogenic cycle, that the certain genes can be transferred to the other bacteria, called the vertical transfer. So for the bacteriophage therapy, we use bacteriophage that do not have this lysogenic cycle. That phage have only this lytic cycle. So that vertical gene transfer probability is quite low. It's not 0%, but it's quite low. So you don't have to worry about dissemination of the resistance gene or virulence gene when you use this lytic cycle phages. Okay?
교수님께서
Now, Professor Yu, you have said that the Korean MFDS should be more brave. Well, I'm not in charge of approving such products, but I will do my best. Maybe we can provide you with a team of product support. And yes, we are becoming braver and courageous. So yes, so thank you very much for that comment.